first of all, who are we? And I thank Joe Hopman for introducing the Appalachian Center for Ethnobotanical Studies. ACES is an interdisciplinary, interinstitutional collaboration. It's located at Frostburg State University, where its headquarters are. And it's a partnership between Frostburg, the University of Maryland, West Virginia University, and most recently, Thai Sophia Institute. And the mission of ACES is, first of all, to conduct multidisciplinary research on the conservation of medicinal plants, to foster economic growth, as well as to preserve Appalachian culture as it relates to wild plants and herbal medicine through outreach and educational programs. So why are we interested in establishing a medicinal plant consortium? Well, first of all, as I don't have to explain to this audience, the Appalachian region is very rich in its biodiversity, and plants have long been considered valuable resources that provide substantial economic development or benefit to the inhabitants. We were wondering if we couldn't increase and expand the economic benefit by developing a strategic plan for the sustainable cultivation of medicinal plants. So why medicinal plants? Well, first of all, 25% of all the drugs that are currently prescribed derive from plant-based material. And also, at least half of the United States population use some form of botanical supplement, with medicinal plants constituting the fastest growing segment in that sector. Medicinal plants clearly have some therapeutic potential, but there are also some adverse effects, and that's particularly the case when they're available in an uncontrolled manner to an inadequately informed public. And from a scientific perspective, the biological effects of plant materials can vary significantly. First of all, they're poorly defined mixtures. Sometimes we don't understand all of the active principles or their unidentified mechanisms of their action. There's also considerable variation from one preparation to another. And that can be for a lot of different reasons, some of which I'll mention today, and which are worthy of scientific research. So there are a number of medicinal plants in the Appalachian region that we're interested in. Here I illustrate a few of them that we have looked at, including bloodroot, blue cohosh, fairy wand, and golden seal. These are some of the bioactive compounds or natural products that are isolated from these compounds or from these plants. These plants and their extracts have been used since the original settlers in this area for a variety of maladies. More recently, through natural product isolation, compounds like these have been looked at in a number of clinical trials and have shown a lot of antibacterial, anti-neoplastic activities, anti-tumorigenic activities. So there's an ongoing active area of research in terms of natural product development for these plants. The plant that we're going to focus on today, and one that we spent the most time on at ACES, is black cohosh, or Actaea racemosa. So black cohosh is a perennial in the Vernoculaceae family. It grows to about 2 or 3 feet tall in the mountain regions. It's indigenous to eastern North America. It grows from Ontario to Alabama. It's long been used by Native Americans. In fact, its use predates the European settlers to treat a variety of issues from rheumatism, malaria, and infections. The business part of this plant, the aerial part is quite interesting. It develops these interesting and quite striking white racemes that have a rather pungent odor. But the business part of the plant are the roots and rhizomes, which are dark colored because of the high level of phenolics that they contain. And these are typically used in a lot of different mixtures as a sedative, analgesic, or anti-inflammatories. But today, the most common use for black cohosh is as an over-the-counter botanical dietary supplement, a multi-component botanical therapeutic, if you will, that you can buy at any drugstore or supermarket without a prescription or ID even, that's marketed under a number of names to ease menopausal discomforts. So it's readily available, widely used, but the mechanism of action is not well determined, and there are a number of issues with authenticity and its mechanism of action that we'd like to investigate. So why were we motivated from ACES to begin to think about trying to build a consortium among a diverse group of stakeholders? Well, our field and laboratory studies on black cohosh raised a number of interesting questions about not only the authenticity, but the phytochemical quality of not only black cohosh, but a host of medicinal plants. And with the increased interest in economic viability of wild harvested non-timber forest products or medicinal plants, there's leading to a destruction of plant populations. Alternatively, wild stimulated cultivation is becoming more sustainable thanks to people's efforts like Jim Chamberlain, who's here today and will be speaking shortly. And with additional research, this might be a nice alternative approach to develop authentic, high-quality plants for some economic benefit. So I want to tell you a couple of interesting laboratory results on black cohosh that led us, or that motivated us to think about trying to organize this symposium based on issues about its genetic authenticity and its phytochemical quality. So in our lab, we have about 150 plants, different plants, black cohosh plants, that we've collected from various parts of North America. We've gotten some of these from so-called reputable botanical dealers. Some we've taken citizen science walks to collect some from various parts of the country. We've gotten some from the USDA that are certified germplasm accessions. We wanted to ask the very simple question, can the genotypes of these plants correlate with different chemical phenotypes? So in other words, with the natural products that some of these plants display, correlate with some genetic diversity that we might expect to see in these various populations. So we tried to do a simple experiment where we've amplified certain regions in plant genomic DNA, the internal transcribed spacer, which is a nice region to look at genetic diversity in populations by using PCR. We could sequence that amplified DNA, and then we could analyze with those sequences to compare the molecular genetics of that plant population from the various places we collected them with different phytochemical profiles. So when we first did this, when we initially started to accumulate results, we found that the sequences distributed phylogenetically in a rather striking fashion. We found that plants from Maryland or Virginia, North Carolina, or Alabama were indeed segregated phylogenetically based on single nucleotide polymorphisms. And this was quite gratifying, and we were interested in then looking at some of the phytochemical distributions. But we didn't stop with this first subset of plants. We continued to sequence a variety of the plants in our collection. What we found was something quite striking. In addition to these 4 different phylogenetic clades, we found another very distantly related clade in our population, which wasn't Actaea racemosa at all, but was rather Actaea podocarpa, or yellow cohosh. This was quite a surprise to us, something we didn't expect, and we reproduced this to make certain that some of the plants that we had, indeed, that we thought were black cohosh, were in fact yellow cohosh based on their genotypes. So one of my colleagues pointed out this interesting quote to me from a 1914 issue of the California Eclectic Medical Journal, which says, "Audacious indeed would be the dealer in American crude drugs who would attempt, by their roots, to distinguish between Cimicifuga racemosa, or black cohosh, and Cimicifuga americana." So what we learned from this experiment was that in spite of the fact we got our plants from a variety of reputable sources, looking at least from our point of view, looking at simple botanical characteristics were not enough to distinguish plants based either on their roots or when they were very young and had not been flowering yet on their true botanical identity. So regarding phytochemistry, black cohosh also has been characterized quite extensively by a number of laboratories using natural product chemistry and through phytochemical analysis. And there are a number of interesting compounds that have been identified, including some phenolic acids like this, cimicifugic acid, which is biphenolic and has strong antioxidant properties. 23-epi, 26-deoxyactine is one of the most abundant compounds in black cohosh. It's got a variety of anti-neoplastic as well as anti-osteoclastogenic effects. And more recently, there's an interesting report about the presence of N-methylserotonin in black cohosh. This turns out to be a very strong agonist to the serotonin receptor, and may well be responsible. In fact, it's, I think, the leading small molecule for the effects of black cohosh in alleviating menopausal symptoms. Interestingly, though, the black cohosh that you can get from the supermarket or the drugstore is standardized against this compound, 23-epi, 26-deoxyactine. Not necessarily because it's the most biologically active, but because it's the most abundant compound in that extract. And in fact, as long as that extract has deoxyactine, regardless of whether it's composed purely of black cohosh or it has yellow cohosh or even grass clippings, it can be labeled as black cohosh. Instead, it might be more beneficial to look at a compound like N-methylserotonin in extracts in order to evaluate plants with high degree of phytochemical quality. So one of the kinds of things that we do in our lab is to look at the genetics and genomics of medicinal plants and correlate gene-metabolite relationships by doing a variety of high-throughput sequencing as well as high-precision mass spectrometry. And we've worked out a simple pathway for N-methyl serotonin that actually is going to be presented later this week on Thursday morning by a colleague in the lab, Martin Spearing. So I'm not going to give you I'm not going to steal any of his thunder, but let me just suffice it to say that the way we do this, and Martin will get into the details, is we can clone genes from black cohosh and analyze them in an organism like yeast, where we can readily express them and induce specifically their expression and assay their products using techniques like mass spectrometry. But we can also assay for these compounds in the plants themselves. And what we found, and Martin might explain this more carefully in his talk, is that we can identify these interesting tryptamines, either tryptamine, 5-hydroxytryptamine, or N-methyl serotonin in plants, but we find them in different tissues and at different stages of development. So this calls into question harvesting times, the appropriate plant to harvest in terms of its phytochemical quality, and what might be an appropriate plant to harvest to maximize the type of beneficial effects we hope to engender in providing for the stakeholders. So there are a wide range of scientific activities in ACES, and I can't really describe all of them today. But made a list here, and I'll try to go through it quickly just to give you a sense of some of the kinds of things we're trying to do. So and you'll see posters and presentations throughout the week on some of these projects. So there's mapping the natural habitats of key medicinal plants in the Appalachian regions. We're trying to extend the readily verifiable methods we've developed in the lab to more do-it-yourself methods that can be used at remote locations by stakeholders to either authenticate or to verify the quality of medicinal plants. We're interested in efficient germination protocols for a variety of plants. Many of these plants do not germinate very readily, and it takes some considerable effort. We're looking at optimal resource allocation in terms of how to grow these plants in nursery-like conditions or in forest-like environments. We're looking at a variety of tools, both in the lab using LC mass spectrometry, but also a remote technique like TLC that we can use to ensure phytochemical quality of plants. Some of the things I won't have time to talk about, Martin will reference this, is we're developing laboratory systems to use genomics and metabolomics technologies to really identify the factors that are involved in, that lead to the phytochemical quality of these plants. And then, of course, we'd like to use that information to develop hypotheses so we can begin to think about how to breed plants of improved phytochemical quality. So the activities really are focused on taking a scientific approach to develop and cultivate high-quality medicinals because we think those could lead to greater economic benefit. So with that sort of a background, the goals of our consortium we have some scientific and outreach goals, and that would be to identify the environmental and genetic factors that give rise to plants with enhanced phytochemical attributes. We want to, again, develop guidelines for the economic feasibility and profitability for wild simulated growth and harvest of medicinal plants. We'd like to establish regional outreach programs as well as demonstration educational activities to demonstrate best practices in the growth and cultivation of medicinal plants. Develop community resources that could be used in the field or else in the remote laboratories for genetic authentication or post-harvest quality controls of medicinal plants, and to advance our understanding of a variety of medicinal plants in Appalachia to think about how we might tackle a breeding or developing variants with improved quality. So we're looking for partners. We want to know if you're interested in joining a medicinal plant consortium. We're interested in linking lab and field scientists, as well as growers, harvesters, clinicians, economists, landowners, sociologists, extension agents, and industry to advance the economic potential of medicinal plants. Would you attend a regional symposium? The fact that so many people from so many countries have come to this meeting suggests that hopefully you will. We're trying to plan a symposium where we can look at what the technical, cultural, and economic opportunities and barriers are to cultivating medicinal plants for improved economic benefit as non-timber forest projects. And the question we want to ask is, can we develop a meaningful strategic plan? If so, can we implement that? Or what are the outreach efforts we need to consider in order to implement that plan? How do we want to be able to coordinate the technical and outreach activities to establish favorable conditions and establish stable markets? So if you're interested, the ACES website is kindly hosted by Frostburg State University. Please visit the site. It asks for your feedback, for your input, for your thoughts, for your criticisms. We'd be interested in hearing from you, and we'd be promised to stay in touch. So quickly, I'd just like to acknowledge a few of the people who have been involved in this work. At the University of Maryland, Martin Spearing, again, he'll be giving a talk on the tryptamine pathway in black cohosh later this week, as well as some other folks in the lab. At Frostburg, obviously Mimi, who's the outreach coordinator for ACES and Sunshine, and some of our students, including Amanda Vickers, who have been, we've been collaborating with on looking at the phytochemical quality of natural products in plants from various locations in Maryland. Anna Sano and her two students from Bowie State, Chinea Fraser and Akeen Vaughn, initially started to do the genotyping work. And interestingly, when they got their first interesting SNPs that indicated that some of our plants were not black cohosh, we were very skeptical and thought they would have made nothing but mistakes until we reproduced that and verified for ourselves that indeed we had some interesting contaminants in our culture. And lastly, I'd like to thank the USDA and ACES for funding, and I'd be glad to take any questions if there's a minute.